I want to introduce Andrei Levchenko. If you have not read the paper Chong et al. In Science, it is the foundational paper on how to understand how cells process information. And the only reason why they were able to do it is because they were able to do single-cell measurements. Without single-cell measurements, you can't do this job. That is the paper that I show everybody, that is wondering whether you can actually use information theory to understand cellular signaling. And on top of that, Andrei used to be on the same floor as I in the Moore building at Caltech, so we go back a long time. So he's now going to tell you about information encoding and decoding in living cells.
Yeah, thank you very much. Ladies and gentlemen, thank you. I can go now. After such an introduction, I can leave. I think it's better to leave right now before I mess it up. All right. So thanks for staying, and I'm sure things will be a bit more lively now that we've had a little bit of wine, and so we're sort of recharging the batteries a little bit. And thank you very much to the organizers. I think it's been really an amazing day. It's very interesting to see all these different levels of description, different questions, different approaches. All of this is really fascinating. I really was dreaming about the time when such a thing will be possible. So I'll try to kind of make things perhaps a bit less serious. I don't know, there are a lot of things jumping around here. Oh, there we go. Wow, it's amazing. So I'm going to try to master this as I go. So this is, again, this is the work, as we sort of advance in science, we stop doing science altogether. And so it's mostly cheerleading the super talented students and postdocs in the lab, and being incredibly thankful to the funding agencies, including NSF and a variety of different sources, who really have patience with us and a desire to support some of this work. So you already heard a lot about how there's this idea of information in the cells of two kinds. One is this hard-coded information in the genome that still can change, but perhaps over longer periods of time, and this more kind of fluid, less defined information around the cells, but that's something that really mediates this selection, for example, or inputs that the cells may need to be aware of. So just put it here for a second so it doesn't jump. So we are interested in specifically how the cells really perceive the changes in environment and respond to those changes. And they do have dedicated networks that seem to really be there for that purpose, to transfer information, the signal transduction networks. And so it's very natural to think about the chemistry of that, of course, but also what is what are the information processing properties of those networks. And of course, the other thing you are always trying to, or at least I'm trying to keep myself aware of, is the physiological setting or pathological setting in which cells talk to each other or signal to each other. And so I'm going to try today as much as I can to kind of keep you focused on a few of those, but primarily be motivated by this situation that I think everybody in this room has experienced in one way or another. This is a wound, a cut perhaps. So when you have a cut, you can observe this for a couple of weeks, sometimes for a week, this whole healing process, and sort of like an experiment being run on your finger by your body. And you can just observe that and wonder how all of this is working. And so here, what happens frequently is that if the cut is sufficiently deep, then you'll see that there's some blood, there's some swelling, all of those other things, and that just indicates that the blood, the vasculature, the blood vessels, have been somehow disrupted. And that means that there will be lack of oxygen or nutrients in the tissue, and therefore a need to repair that in some ways, and that's the wound healing part of all of that. The other thing that happens quite a bit, and also mentioned already, you'll have infection frequently. Some sort of foreign bugs or objects will get into the wound through this now disrupted protective barrier. And so there's a homeostatic, in some sense, sort of need here to recover the state that was there before the cut, and to take care of the infection and inflammation is the whole process that takes takes care of that, inflammatory responses, and also rebuild the vasculature. So, the way it happens is that there are these sentinel cells, I'll mention them a couple of times, that are embedded in the tissue. Their job is to sense what's happening and, for example, sense the presence of these foreign objects. So lipopolysaccharide is a part of the bacterial cell wall, and that's something that can be sensed by sentinel cells such as macrophages, one of the most common type. And they will transmit that in a kind of a broadcast fashion through this information, now encoded in a chemical signal that now begins to spread from the macrophages and hits whoever wants to listen, right? Whoever has the right receptor. So, for example, one of these broadcast signals is this already mentioned tumor necrosis factor, just a cytokine. There are other cytokines. And so this is what macrophages will be sending to any other cells, including cells in the blood vessels. Okay, so the blood vessel cells, their job is at this stage is to not just yet to repair it or to kind of grow and reconstruct the whole vasculature in there, all these complex networks of blood vessels, but also open up the windows between the cells, the cells kind of come apart a little bit, and allow the cells that are sort of natural-born killers, like neutrophils, to get out of the bloodstream, which is a huge highway system that we have in our body, to exit from there, to get into the tissue and start killing bacterial cells that may be there. So that's sort of the communication and the meaning, the semantics of that communication through, let's say, tumor necrosis factor. There is another, as I said, need for the cells to do something else, and that is to start growing these new blood vessels. So something that's called angiogenesis, and it's surprisingly the same cell, macrophage, that will also detect a different input, lack of oxygen, and will translate it again into a broadcast chemical signal. And there's a name, VEGF, vascular endothelial growth factor. Somebody already mentioned here, don't pay attention to those letters, right? So the key is just to sort of think about this environmental inputs being translated by sentinel cells, the cells that are there for sensing, and then these cells convert that information into a chemical signal that starts propagating away, hitting a variety of different cells around. And so the way this new blood vessel will begin to grow at some point is by a decision-making here, where that one of the cells in the blood vessel in the wall, there are many cells, will have to decide to become a so-called tip cell, the cell that will lead the way in the growing blood vessel, and cells around that will have to be now to make a decision to become so-called stalk cells. They'll be dividing just behind the tip cell to enable the growth of the blood vessel so it doesn't break away. So the tip cell, as it tries to move out, will not break away completely, so the cells just behind will be dividing. So the decision is some cells will need to be dividing, some cells will need to be moving. Okay, so just to kind of, wow, it's a very sophisticated system here. So basically this is the idea. You have a couple of different inputs that are sort of of physical nature being translated by the macrophage, the sentinel cell, into two chemical outputs that hit the same cell, endothelial cell, that lead to two different actions by endothelial cells. First enables taking care of the infection through recruitment of these immune cells, and the other one to growth of endothelial cells for decision, to start somehow this complex process of growing a new blood vessel. So there's a sensing and there's an execution, already mentioned also today. And the way the stalk and tip cell definition here takes place is by also the cells talking to each other. They're not just independently deciding who's going to be the tip cell and who's going to be the stalk cell, but they actually are doing that because they're next to each other. So they're talking to each other for a while. So this is the overall system that I'd like to kind of talk a little bit about today. And so you have VEGF, this signal that promotes growth of new blood vessels, affecting two cells that are, to begin with, appear to be either identical or kind of naive. So they don't know that they're going to become tip cell or stalk cell necessarily. Somehow they figure out what they will become, and that will, if that is done successfully, this will lead to growth. Now, going back to TNF, I implied that this is specific to taking care of the inflammation of or enabling inflammation, but that actually does affect also this outcome, the angiogenesis or growth of the blood vessel outcome. In fact, if the concentration of this inflammatory signal is high enough, the blood vessel is not going to grow, right? So inflammation, this infection needs to be resolved, taken care of first before the blood vessel growth begins. So this is now I think it's a complete system. I should say that, in a sense, you can see it as a part of this kind of loop of homeostasis loop, right? You have some sort of input, and in many cases in physiology, it will be a deviation from normalcy, right? So for example, here, lack of oxygen, presence of bacterial cells, it could be presence of glucose. Sentinel cell there will be beta cell in the pancreas that will translate that into insulin secretion. So again, translation of an input that would be external to the system, glucose, to the output that is a chemically induced signal, and so on and so on. There are many such examples. And so this input has to be somehow passing through some information channels that we discussed today, translated in some growth of the blood vessel, which implies, again, movement and cell division, could be death. We already heard about that from Eric, and I'm going to mention that again if I have time, and so on and so on. And that ultimately takes care of this input, right? So there's a bit of a control theory aspect to all of this, which is an interesting challenge, how to combine what we've been talking about today with the control theory. Yeah.
What's the logical big box?
Say again? Sorry.
Logical big box.
Oh, the big box is, I have no idea. Forget about the big box. All right. All right. Maybe there is a meaning to it, but we haven't found that yet. All right. So again, this paper has now been mentioned so many times that I'm not going to even say anything other than that when you look at this information channel, it doesn't need to be this particular pathway. In one way, even though there is sometimes a huge chemical complexity to this process, you can really establish an input and output and look at it as a kind of wire. That's something that will transmit information subject to some noise. And one of the questions that has motivated us, but then a lot of people after this initial work, is can we sort of follow the idea of quantification of this information capacity? It's very hard to establish what we would call sort of what could be done, for example, in Drosophila. It's already been mentioned today by Tkachik and some other people, where you know the input and output. Here, in many such cases, it's very hard to measure the input distribution. So the best you can do is to measure capacity. In other words, you find this input distribution that maximizes the mutual information, and then you take that maximal value of the mutual information as a kind of characteristic of this communication channel, subject to specific noise. So most of the paper, even though the idea that it's such a low capacity channel was very, very striking, a lot of discussion after that was, again, how do you take care of this? How do you potentially improve that? And there are many strategies you can follow. A lot of those strategies rely on integration, either integration over time, integration over multiple channels, or integration over multiple cells. Multiple cells allow you to kind of get away from the box of the cell, because inside a single cell, even if you have multiple parallel pathways, they all can be subject to information bottleneck or some reduction of information very high upstream, which frequently does happen in biological systems. But when you have multiple cells, you are not frequently subject to that, and it's a very powerful strategy of transmitting information. One interesting aspect of I'm going to come back very briefly to it in a second, but it's even though you can look at it as a single pathway, you already begin to see some network aspects when you start looking at this a little bit more closely. So TNF can stimulate signaling over multiple pathways, and they seem to be branching out. And so you have this rather branch structure to this network. And then you can look at a variety of transcription factors that are stimulated by the same input. And what you do find is that the outputs are correlated quite a bit. And that's one of the reasons you really have trouble taking advantage, in addition to the information bottlenecks, some noise that can be introduced very early before the branching happens. In addition to that, this correlation in terms of the outputs kind of makes it very difficult to use them as independent channels of the information. So we'll come back to this, but one of the questions that I wanted to really emphasize today is how do you also use the information, right? So right now we have this channel, there's some information flowing through the channel. is it used in certain, in any way, and particularly because, this is a, kind of not a lot of information. So what can you do with that? One thing that already was mentioned today by Tom and others is that one useful way to look at it is these distributions. I already showed you kind of two-dimensional distributions. But back to one dimension, you can compare, for example, weak and strong stimuli and see how well resolved they are or not so well resolved they are. And the difference between, for example, positions of the peak would be your signal, and the variances, or sort of how broadly this distributions may be compared to that difference between the peaks, will indicate how noisy. And so the signal-to-noise ratio is really that difference between the peaks versus this variability. And so that's quite useful because you can use some of these concepts that have already been mentioned for information analysis, but also kind of begins to tell us something about the decision making, right? So if you have maybe approximately 1 bit of information, so what does that mean? It characterizes the channel, but also characterizes what you can do with the channel. So one thing you can do with the channel is perhaps enable some sort of binary decision. You can say, well, maybe the cell will decide between surviving, dying, becoming tip cell, stock cell, doing many other things that we mentioned today. So, there's this threshold that Eric Dietz mentioned might exist, for example, when the cells decide whether they live or die. He didn't tell us a little bit, a lot more about where the threshold comes from. So, but it could come from something, downstream, as they say, or where this information flows, some sort of a different circuit that enables that. So, with that in mind, you can ask a couple of questions here. One is you can look at it as a potentially interesting decision-making kind of example. So in decision-making, for example, you're trying to detect a plane and distinguish it from the rocket. Apparently, some people do it well, some people don't. And so you kind of either shoot it down or you don't, right? So this is a very frequent problem, classification problem. And so what you don't want to have is error, right? So if there is no rocket, there's a plane, for example, you don't want to trigger response. Vice versa, right? Sometimes you don't think that there is a rocket hits you. You may want to also pay attention to that. So there's a sort of 2 types of error that you'd like to minimize, so-called miss and false alarm. They're related to false positive, false negative type of errors that people do talk about frequently, but this is also about this overlap. So when you try to optimize the decision-making classification, you would like to put it so that you minimize overall error, right, both parts of the error. And that will be, in many cases, considered to be kind of where to put that threshold for detection of this rocket. Or for cells to really sort of this is how we're going to make that decision, binary decision, to do something or not. But it's not very clear if at all times this consideration of optimality of such a decision is what actually happens. And it's not very clear where, again, this threshold comes from, all these other things. So, so to think about this a bit more, let's, again, sort of revisit what we just discussed. So we have perhaps one-dimensional, two-dimensional outputs. It's very different from the usual sort of idea of one input, one output. Have some clouds, some distributions of responses, perhaps in 2 dimensions. And as input increases, they kind of shift. So, so all of that is unstructured, so to speak, right? And so the question is, maybe there is something else to this diagram that we can put in. And this something else, again, will come from where this information flows, from this execution, so-called execution modules, the way we call them. And again, this is this idea of specific circuits that may exist for triggering cell division, migration. They are separate from signaling, but they are modulated or biased by a signaling process, right? And they have their own property. In fact, frequently you'll see this feedback-rich kind of modules controlling cell division, other things. And arguably, right, this is what can structure the space that we're talking about. So, for example, a positive feedback will give you 3 possible outcomes, low, high, bistable, frequently. Positive and negative feedback together give you a similar kind of almost like phase diagram here, where you have, instead of bistable, sometimes you have oscillatory response, for example. And so you have this type of structure that may exist in space. And so when you think about this on the left, what we've been talking about, signaling. Now signaling, this output distributions or clouds get superimposed onto this kind of now more structured space, right? Provided that these outputs that we talk on the left will be connected to some aspects of this execution, right? So for example, output number 1 may affect parameter 1, output 2 may affect parameter 2, and so then you can really map whatever we have on the left onto this right-hand diagram. Now, Eric already talked a little bit about the decision to live or die. Now, this is one of the simplest decisions in some sense because it does depend, for example, on this so-called pro and anti-death proteins. And if you have about the same amount of them in the cell, then the cell kind of sits on the fence. If you have more proteins that will enable death, the cell dies. Less, the cell survives. And so this really translates in a very simple linear diagram. We have two kinds of proteins, and there's some line that separates this life and death situation. In fact, in individual cells, you can measure distributions of those proteins, and you can do it both in the health and in some weird state, in this case, epigenetically controlled state. And that allows you, again, to take these distributions, or even distributions of signaling inputs that will control those proteins, pro and death, and map it onto this diagram. So there are a few more examples, but I'm not going to go through them, of how you kind of do this type of mapping, but I'll be happy to discuss. So let's go back to this system that I described in the beginning, this growth of blood vessels and resolution of infection. So first you need to resolve the infection, and then you can grow this blood vessel. So one example is shown there. You have this existing blood vessel below, and there is this new so-called sprout emerges from that. And so how does it happen? So again, let's go back to the idea that I showed you in the diagram before. So you have two cells, they're exposed to this input that triggers this separation of fates. So one cell starts moving, the other cells around start dividing. Overall, there's a sprout growth. And so there's a lot of interesting information there that you can embed into analytical framework, and again, end up with this, again, phase diagram. In this case, you have two inputs that are of interest to us, one of which is VEGF and the other TNF, these two signals that we show here. And the population would be expected to be somewhere here, right? So based on what we know about TNF and how it gets into NF-kappa B signaling that I showed you before, there's going to be some distribution, and there's going to be a sharp boundary, kind of, as it turns out from this analysis, from this process of discussion between the cells and ultimately differential fate decision. There's a pretty sharp threshold coming from there that would separate no differentiation, the cells remain undifferentiated, kind of the way they were there, and differentiation here. So the problem, we don't know at this point if it's true, but we can at least structure that space onto which this input from or output from signaling will be mapped onto. Now, we did this analysis in the lab where we actually built our own. So this is a device where it allows you to kind of grow these blood vessels, right? And we can also simultaneously control the inputs. We can add our own TNF and our own VEGF, these two different inputs, and in fact, really different combinations of those. We can really sort of go wild on combining them. And that, first of all, allows you to really track. First of all, you obviously need to convince yourself that this overall idea of this threshold, right, being very sharp, and if TNF is very high, then it kind of puts it above the threshold, and that means no blood vessels grow. If it's lower than that, it actually doesn't prevent that from happening. But what was interesting is, as we were sketching out all of this, we found that we can really land onto this threshold. In fact, we can sort of be very, have these input combinations that will have very different outcomes, either completely zero blood vessels or a lot of blood vessels, right? So this is, for example, shown here in 3 and 3 prime, the way we call it. These are two experimental conditions, but they're actually identical. What happens is that either you fall on one side or the other side of the threshold, but essentially you're sitting, you're kind of, and this is the only case in all of these experiments. In fact, we have more, only when you're on this threshold you have this either essentially just tip cells forming, but not real blood vessel, right, or extensive blood vessels forming. So what's interesting is that on the one hand it's nice to have this, right? On the other hand, you start wondering, well, how come? What I just told you is that there's a lot of variability in the system, and yet the system somehow can decide so well to either form blood vessels or to immediately shut it down. In fact, with this incredibly sharp threshold. So what happens there? Why is it not doing kind of this variable, input-output relationship where you get, a lot of blood vessels or maybe a little bit less blood vessels? We've heard a lot about how noise, for example, can let you get this more kind of gradual response, right? So this population, if it gets superimposed, this cloud, this distribution, should give you kind of a lot of different examples, even for the same set of conditions, input conditions, right? So maybe there are two possibilities. One is you actually put a bit more premium. You shift the threshold, decision threshold, from somewhere in between where you minimize both kind of this miss and outcome. When there's something and you miss that, right? Maybe you want to minimize that, and you don't care about the other error, right? Because if you want to prevent something from happening, you really want to worry about missing the input. And if the input is there and you're not responding, that will challenge your your fitness, in the language that was used today, quite a bit. So you can shift that. The other thing that can happen is this. So here again, we are back to this multicellular processing. Okay? So multicellular processing, we already again heard from Eric that it could be fractional, right? So you don't care which cells do what, you just care about 50% of the cells doing one thing and 50% of the cells doing something else. So here we have a slightly different example of how multicellular processing can happen. And again, this is because if you look again at the existing blood vessel, for a new blood vessel to form, you kind of need to have a collective decision where it's not just even sort of two cells, as I showed you in the diagram before. It's actually a number of cells around a future tip cell has to decide to become stock cells. And none of them should be shut down by TNF. In other words, if TNF input is there, or even if it's not there, it should be that all the cells, right, are interpreting the outcome as if they don't have this problem with the missed outcome. They essentially have to be in the right state, right? So you kind of need to find this group of cells, all of which together, right, made a decision that the input is below the threshold. So that means that for each individual cell, right, there is a cloud, there's a pretty wide distribution. But if you start figuring out, right, what is the probability when the cells have this outcome, what is the probability that they'll interpret that the signal is not there, which is what we need here, and then you multiply those probabilities, you'll get a very sharp decision threshold, even though all of this, again, is sort of each cell makes their decision independently, in principle, and then they can start interacting with each other. The decision allows them to interact and ultimately figure out what to do. So overall, and I'm finishing, overall, I think there are a few interesting things that we've talked a little bit about today. One is, when we talk about, in fact, our paper in the beginning was really, we have information or capacity to transfer information, essentially think mostly about what happens in a single cell. And there are many single cell sensing response cases. We've heard about bacterial chemotaxis, for example. A single cell may be kind of trying to make a series of decisions. And there is a certain certain important properties of that. We already have heard that, a little bit about that from Christoph. But overall, the cells need to kind of be able to look at the same input, right? So the only way they can integrate here is over time. And yet, in a sense, they shouldn't have a long memory of who they are. So it's a bit hard to reconcile that. But one way they can integrate over time is by actually moving, right, in space. They record, the information is recorded in the overall trajectory. When we look at the position of the cells, we actually look at how they accumulated all this information in different steps without necessarily even having chemical memory. So it's a rapid randomization, allowing them to look at the same input in different ways and also kind of try to record that over a certain amount of time. Now we have collective cell decision. There could be a coordinated situation where the cells may exchange the information. They all may, in fact, even independently get the information, but they all need to arrive at the same decision. And it's a little bit easier for them to do that again because you still have integration over multiple cells looking at the same signal. Or it could be fractional. This is what, again, Eric was talking about. The only condition for that, okay, sorry, might have lost it. The only condition for that, I don't know if I lost it completely now, is, I think I pressed something. Oh, there you go. The only condition for that is that all of the cells need to kind of be exposed to the same signal. And we've heard already that, if you have 2 cells, fractional decision making obviously doesn't make sense. If you have 10 cells or 100 cells, it starts making sense. But they all should see the same input. So it's an interesting thing to, I guess, discuss, for us to discuss. And also the fact, again, if you put it all together, we talked about information channel properties. We talked about phenotypic programs that are already pre-existing in a cell. And you're using information to kind of bias the decisions one way or another. But you don't inform the cells about the complexity of decision making. That's already in there. You just need to map onto various programs and try to bias them depending on the actual value of the signaling outcome for the cells. And all of that, in many cases, will be a part of the loop that might be also interesting to think about from the control perspective, because frequently it will be in a homeostasis situation. So a few of the questions that I think might be interesting to discuss or think about or research into is how this idea of homeostasis and many different of the signaling processes, how this idea of control of theory kind of fitting in all of this, how all of this can be combined. How do we describe all these pre-existing cellular programs? Can we have some coordinated response where many of them get sort of activated in some coordinated way? And how these binary decisions that may be enabled by a single bit of information passing through the channel can be refined. For example, you can create these very sharp thresholds in spite of the fact that it's a very widely distributed response. How you can take advantage of these collective responses. Finally, I think it really, what we haven't discussed much, but we started looking into here, is how do you deal with multiple inputs and multiple outputs, right? So this MIMO idea that obviously is also quite widely discussed in computer science or in other settings. How can this be used in the biological systems? Thank you very much.
We don't really have, we have time while we're changing computers, so.
So just a very quick thing, Andrei, but you can optimally shift your thresholds in binary decision making by ascribing a cost to each of the both correct and incorrect decisions. So and computing that modified threshold is actually quite straightforward for Bayes optimal detection.
Right. So basically, my point is the following. So there are a couple of different sort of levels here. One is what you would sort of associate with information channel, and the other is this so-called execution, pre-existing ability by the cell to move, to divide, to do other things, right? So you kind of connect the two. Now, if you perturb the execution module, you can shift your thresholds all you want, right? It's really a function of what happens, what is pre-existent program, but in biology you can change that if you want. But if you don't change that, then it may be somehow there, right? What would be adjustable is the input and the movement of distributions in a structured space, and the space is structured according to those thresholds that pre-exist. All right. So then you kind of can move and explore different behaviors.
All right. Let's thank the speaker again.